Welcome to my bachelor thesis defense. My bachelor thesis is titled Unsupervised Learning with Applications in Genomics. This project has been carried out upon the collaboration of Universitat Politècnica de Catalunya and Stanford University with the advice of Drs. Alexandre Ioannidis, Daniel Mas Montserrat, and Xavi Girón Neto. Today's presentation will be organized as follows. First of all, I will explain the motivation behind and the value of the project carried out, as well as the important issues that it tackles. Then we will go with the proposed method, and I will show the experience and the results, the promising results that it shows. And finally, we will conclude with some final thoughts and comments. So let's start with the motivation. And to explain the motivation behind this project, I will explain you how current medicine does work. Imagine that we have a group of patients and all of them have a certain disease. Diseases have generic therapies, and those therapies may work on some, may have no effect on others, and in the worst case it can have even adverse effects on some patients. Have you ever wondered why can some drugs have adverse effects on some individuals? This has a biologically simple explanation. Drugs are metabolized by specific enzymes, and each enzyme is determined by the genome of the individual. Therefore, the drug response is directly dependent on the genome of the individual. This is where precision medicine comes to the play. Precision medicine aims to maximize the effect of drugs given the particularities, the characteristics of the genome of an individual. And precision medicine is the future medicine. And the paradigm is a bit different. Having the same set group of patients with the same disease, first of all we will analyze the genome of each individual, and we will find groups of patients which would require a similar therapy, this way maximizing the effect of the therapies. And there are two actors, two phenomena that play an important role in all this process. We have pharmacokinetics, which is how an organism affects a drug, and we have pharmacodynamics, which is how a drug affects an organism. And the thing is that there are significant differences not only between individuals, but also there are differences at the population level. And this is where we have to find out or determine what is exactly a population. Just to give you an example, ibuprofen, which was the precursor of ibuprofen, before ibuprofen in the market we had ibuprofen. Ibuprofen worked quite well in the Japanese market. Worked quite well means that we had a relatively small proportion of patients which had adverse effects. However, this was not the case of the UK market. In the UK market, this proportion was quite large. To the degree that it was dropped from the market because it gave to many patients a liver toxicity. And later on it was dropped from the world market, and ibuprofen came to the supremacy of this setting. Concluding, interethnic differences are crucial to establish health policies. When I say interethnic, I don't mean racial. Racial identities have been established culturally or historically. However, interethnic differences are natural genetic variations present in the genome. And that is why that we cannot say that ibuprofen does not work on Japanese, but it works on English, because if we have a not mixed individual, a child of a Japanese and English, here we cannot classify that, because it is not so straightforward. We cannot stick to racial identities. We have to have a precise definition of the genetic variation. That's why the first task of the project is to classify individuals into natural, not culturally established ancestry groups, into natural ancestry groups or regressing their geographic origin. With this problem in mind, we have another challenge. When we sequence is determining the nucleic acids of the sequence of nucleic acids of the DNA of an individual. Sometimes we have sequencing errors, which induce missing values in the sequence. This is a problem because for precision medicine, we need to know what are exactly those positions if we want to define the ancestry. And that's why we need good imputation tools. Imputation means that we fill in missing values. So this will be the second task we want to address. If we start sequencing lots of sequences of many individuals, we end up having large amounts of data, and that data is really high dimensional. We have thousands of positions. This would require large amounts of space for storage and large channel capacity. And I think you can guess that this is a problem of storage. We need compression tools which can exploit the features of this specific data type. So this would be the third task. And finally, the last task we want to address is the missing diversity problem. The proportion of participants in genetic studies is when you give your genome to some databases so that researchers can study. In the public databases, the proportion of participants is biased towards European ancestry people. And this is due to several factors, but an example is that some communities are reluctant to participate because of privacy or some possible misuse concerns which have happened in history. And another problem is that many genomes which are from private databases are protected by those privacy restrictions, and I couldn't share them with some collaborator from another laboratory because I may be breaking the law. So an ideal setting would be to simulate realistic genomic data to overcome the problem of unbalanced ancestry groups. This way we would train models which are not biased towards a specific ancestry. Summarizing, we have 4 tasks. We have classification, imputation, compression, and simulation. The question is, can we have a unique method that can solve all of those tasks? And the answer is yes, my proposed method does that. Let's see how it was defined. What do we have? We have high-dimensional genomic data. We have 4 tasks to solve. Can we exploit any feature of the data to address these tasks? After an exhaustive exploratory data analysis on genomic data, I found out that genomic data has redundancies and has spatial correlations. If data has redundancies, it means that the information is spread over the observed factors, the observed variables. But we could find another space, we could map that data to another space of lower dimensionality, where we explain the same information but in other hidden or latent factors which contain just information and discard the redundancies or noise present in the observed data. So we just want to find the representation of the gist of the essence of the data. And if we find factors of lower dimensionality, we can solve directly the tasks of classification and compression, and I will show you that they can also be used to solve the tasks of imputation and simulation. And remember those 2 colors, green and pink, because I will use them in the next slide. What do we need for that? We need a parametric model. And in this case, I have chosen the autoencoder architecture, which is a model of a hourglass shape. We go from a high-dimensional input, we map it to the bottleneck of lower dimensionality, and then we try to reconstruct again the input. The encoder, since it reduces the dimensionality of the data, we are compressing, and we can also classify in the latent space. And the decoder, since we're going again back to the original dimensionality, we can simulate new samples, and we can impute using the spatial correlations which I mentioned earlier to fill in the missing values. And a note about all that is that I will be enforcing this bottleneck to be Gaussian. I will explain the benefit of that a bit later. And the whole model has been named VAEs, variational autoencoder for genomic applications. Let's go to the experiments by task. This is the setting of compressing, of of going from high dimensionality to low dimensionality. Having a single ancestry strand, the encoder maps this individual to the latent space, and observe that the latent space mirrors the real-world geography. We can find the European near South Asian West Asian, the Africans lie a bit apart. If we have an individual of a non-ancestry, what we can do is just forward this strand through the encoder, and we would find some coordinates in the latent space. A straightforward way to classify this individual is just take the closest ancestry cluster. So we compute the distances to all the centroids of each population, and we assign the label of the closest cluster. Another way of classifying will be using the decoder. So if this individual is African, we expect that the decoder conditioned on African ancestry would yield the best reconstruction of the input. So we end up of having several methods for classification, and the last one is taking the ancestry label of the best reconstruction. Here in the table, don't be overwhelmed. Let's see what are the values on the rows. This is the classic method. We compare with PCA, with the classification of the linear space, PCA, which is the classic method for global ancestry inference. Only encoder is the first method, is classifying in our VAE space, and encoder plus decoder is the last method I explained. See that we have larger accuracies for across all populations with the VAE space, much better than the linear classic PCA case. For example, for test data in classification for all populations, we have 74% of accuracy, whereas in the VAE condition case, 87. So that's an improvement. In the imputation application, we will be using the whole network. So imagine that we have an input with missing values. We forward it through the whole network, and the decoder, taking advantage of the spatial correlations which I mentioned earlier, it has been trained to impute the missing values. And here in this table we can compare this learning. So the VAE filling is the imputation with our VAE, and zero filling is the imputation with the most frequent value, which is 0. So that's why it's called zero filling. If we have a strand of 10,000 positions having 75% of missing values, the VAE filling allows a reconstruction of 19%, whereas zero filling just 92. So that's a good result also. We can see that the VAE can actually learn the correlations of the strand. Compression also follows a similar setting. We are using the whole network. We're storing two things. One is the latent representation, the coordinates in the latent space. And also we will be storing the residual, which is the difference vector between the input and the output. Why are we storing the residual? Because if we just store the latent representation, the coordinates in the latent space, that would be lossy compression, because the decoder, it makes errors unavoidably. So we would reconstruct the input but with some errors. But in precision medicine, we need to be precise. So we need lossless compression. And this residual vector allows fixing the errors that the decoder does. So having a lossless compression, the performance of this compression algorithm is computed as follows. We compute the compression size, we compute the size of the residual plus the size of the latent representation in bits, and if it's smaller than the size in bits of the input, then we have a successful compression. Don't be overwhelmed with that table. Let's see what are the values. Each cell represents the compression ratio. If it's 1, it means no compression, no expansion. If it's larger than 1, it means that it expands the sequence, so that's a bad result. And if it's less than 1, so if the cell is green, it means that we have compression. a compression of 0.5 means that we have compressed 2 times the original size. And this is the size of the bottleneck. And here we find a trade-off. If we have a small bottleneck, the residual is non-sparse and it weighs too much to have a compression. If we have a large bottleneck, the residual is sparse, yes, but the bottleneck weighs too much to have compression. So that's why we need to find an intermediate point where we have a relatively small bottleneck and a relatively sparse residual, which we found that the best combination is having 64 latent factors, where we achieve 2 times compression in the European population and in the Native American population. And it can even compress the African population, which is the population with the highest variability. And finally, simulation. Recall that I told you that I'm enforcing the bottleneck to be Gaussian. We're enforcing it to a known distribution because if we know the distribution, we can sample from it. But if we don't know the distribution, we couldn't sample from it. Since we know that the bottleneck is 0 mean unit variance Gaussian, we can just create a random vector, a random Gaussian vector, and use the decoder conditioned on the ancestry we want to create a simulated individual of that specified ancestry. Simulation has been assessed with entropy analysis. If you want to see further details of the entropy analysis, you can check the report. Here I'm just showing the global result. This is the PCA on founders data. Founders data is the original data, the real samples, real individuals. And as you can see, we have some imbalance here because we have a small number of individuals for the Oceania population. However, with our method, with our simulation method, we can generate so many individuals as we want across all populations, and the distribution is preserved in the space. This is the PCA on original data, the 10,000 position data, not the latent space. So simulation results look quite promising. To conclude, this work has been a symbiosis of deep learning and genomics, and we have been able to benchmark VAE-based ancestry classification versus classic PCA-based ancestry classification, and we have proved that our nonlinear approach works better than linear PCA-based approaches. We can also successfully impute genomic data with our denoising VAE. We have provided an efficient and fast method, much faster than classic algorithms for simulation for genomic data. And finally, the best result which we have got is in compression. We developed a novel VAE-based lossless compression system for genomic data. All these findings have been presented in this abstract, which was submitted to the American Society of Human Genetics meeting 2021. So we will expect some news on that. And that would be all. If you have any question, I'm all ears, and thank you for your attention.